what can we know about biomolecules? So today's talk is the introduction, and I will show some examples what kind of study is going on. Okay, here is a diagram. IR absorption, Raman, hyper-Raman spectroscopies. You know, IR absorption occurs when we irradiate the IR light to the molecule. Then the photon energy is converted to the energy of the molecules, and the vibrational state and vibration quantum number increases 1. So this is IR absorption. In the case of Raman scattering, we irradiate the light, then scattering process occurs, then the light propagation direction of the light changes, sometimes wavelength changes. So excitation wavelengths and scattered light. Energy difference between these two lights correspond to molecular vibration. If we tune the excitation wavelengths to real electronic absorption band, that is, when we choose the energy of the excitation beam that matches with the energy difference between ground and excited state, if there is a real state, then the scattered light intensity becomes much stronger. This is the resonance Raman effect. And already we have formulated this process, right? And the third one is hyper-Raman spectroscopy. Hyper-Raman spectroscopy is also the scattering process, but the event is different. In this case, 2 photon excitation and 1 photon scattering occurs. In the case of Raman spectroscopy, 1 photon excitation and 1 photon scattering. In the case of hyper-Raman spectroscopy, 2 photon excitation and 1 photon scattering. The resonance effect is also expected. So by choosing the excitation wavelength whose energy matches with the real excitation of the molecule, then hyper-Raman signal is enhanced. This is resonance hyper-Raman signal. You know, all the methods are used to get the information about molecular vibration. Okay? If our purpose is to get information about the molecular vibration or vibration frequency, IR and Raman may be sufficient. It covers a wide range. But using hyper-Raman, as we see, it has a difference in the selection rule and resonance conditions. So we expect by using hyper-Raman spectroscopy, hopefully we can get new information about the molecules. Not only the vibrational state, some new information. That is the expectation. And probably yesterday, I introduced some examples to apply hyper-Raman spectroscopy to small molecules. Then in the case of benzene having high symmetry, hyper-Raman spectroscopy has a unique selection rule. So hyper-Raman active mode covers this range. So for these B1U, B2U, E2U mode has not been detected using IR and Raman, but hyper-Raman spectroscopy can detect these modes. Okay. This kind of symmetry, discussion of the symmetry is very meaningful if the molecule has a high symmetry. But in the case of biomolecules, most of the biomolecules do not have such a high symmetry. They are mostly classified into C1 group, the lowest symmetry. There is no symmetry species. In such a case, this kind of beautiful discussion is unexpected or probably impossible. Because in the case of C1, all the vibration modes are IR active. All the vibration modes are Raman active. So probably we can detect all the vibration modes using these two. But anyway, I want to know if I can get new information about biomolecules using hyper-Raman spectroscopy. This is the purpose, motivation of my study. So far there have been almost no information about the hyper-Raman spectrum of the biomolecules. I started to examine amino acids first of all. Okay, let's prepare the aqueous solution of L-alanine and set the pH to the neutral pH, set the NaCl concentration. Please be aware, when we study biomolecules, we should control the pH value and NaCl concentration. So this is a measurement at neutral pH. Blue spectrum is hyper-Raman spectrum. Red spectrum is Raman spectrum. Okay, I put together vertical axis is the intensity, horizontal axis is the wave number. Okay, is there any difference? Intensity is different, right? Blue spectrum exhibit larger signal. This large signal in Raman spectrum is small in the blue spectrum. But as you see, the vibrational frequency is the same between the two results. It's natural. Hyper-Raman spectroscopy is a method to detect molecular vibration. Raman spectroscopy is also. So they detect the same vibrational mode, normal mode. Only the intensity is different. So I found in the case of L-alanine, hyper-Raman spectrum is nearly equal to Raman spectrum. Okay, this is the first step. Anyway, we have to examine. Then I change the pH value. I prepare 3 samples at pH is 11.1, 7.4, 1.5. So I change the pH. Then the appearance changed in the pH dependent manner. It is expected because alanine has 2 ionizable groups, COOH and NH2. Okay, at a basic condition, NH2 must amino group must exist in NH2, and carboxyl group must exist in CO minus. And at neutral pH, it must be Twitter ionic form. pH 1.5, amino group is expected to be protonated. This is also protonated. This is an expectation and already accepted idea. Then what is the difference of the hyper-Raman spectrum? So bands indicated by the blue box is related to the amino group. So depending on the change in the NH2 to NH3 plus, a peak disappears and new peak newly appears. Green are the CO minus and COH. Okay, so as expected, peak position is the same between these two conditions because CO minus does not change, so they appear equally. And at base acidic pH, it disappears and moves somewhere. New peak of COH should appear here. Yellow bands are attributed to CH or CH3 or skeletal mode CCN. So anyway, we can understand the results of the pH dependence of the hyper-Raman spectrum by considering the three groups. Okay? As expected. I don't think this information is new. Already we know vibrational peak assignment because it is similar to the Raman spectrum. So we know the vibrational peak assignment, and as expected, based on the structural change, some peak disappears and appears. Okay, so in this sense, this result is the expected result. Still now, I don't think hyper-Raman spectrum gives its unique information. It is comparable with Raman spectrum. Okay, I skip this page. Assignment is given here and entirely matches with those of the Raman spectrum. How about L-asparagine? Black is the hyper-Raman spectrum, red is the Raman spectrum. Okay, in this case, some difference appears. Hyper-Raman band appear here, but there is no peak. Hyper-Raman band appears here largely, but there is only small peak. Raman bands do not appear in the hyper-Raman spectrum. So in this case, hyper-Raman spectrum is not identical to Raman spectrum. So somehow we can expect new information from this result, hyper-Raman spectrum. L-asparagine is a structure of L-asparagine is shown here. So it has functional groups CONH2. There is another amino acid having similar structure, CONH2. This is glutamine. But in this case, surprisingly, the spectrum of the hyper-Raman spectrum, black spectrum, and Raman spectrum, red spectrum, was not different very much. They were similar to each other. Okay, so about this result, we can expect some new information about large peaks. that only appears in the hyper-Raman spectrum. Here is another example of the amino acid. This is hyper-Raman spectrum of L-tryptophan. Yesterday I explained tryptophan is sometimes focused on by using resonance Raman spectroscopy because it exhibits UV visible absorption. Okay, so we measure the hyper-Raman spectrum by 532 nanometer pulse laser. Considering the hyper-Raman process, 532, then signal should be nearly 266 nanometer. Okay, that is, in other words, the excitation energy is 2 photon of 532, so it's equivalent to 266 nanometer. So even though this is a 532 nanometer excitation, it resonates to the electronic states at this position. And at 266 nanometer, there is an absorption of the tryptophan. Okay, this is a tryptophan spectrum. And at 266 nanometer, it has an electronic state. So we can expect here is a real state near 266. So it's possible to expect a resonance effect, resonance hyper-Raman effect. Then we compared this hyper-Raman result spectrum, 532 nanometer excited hyper-Raman spectrum with 266 nanometer excited resonance Raman spectrum. So here is the 266 nanometer excited resonance Raman. This is 1 photon excitation. This is 2 photon excitation. And let's compare this spectrum and blue spectrum. You see, here is the triplet bands. Here is the triplet bands. And this pattern matches with this feature. At 800 wave number or less, there is a peak. At 800 wave number or less, there is a peak. Nearly 900, there is a peak. 1000, there is 2 peaks. So, at a glance, these two spectra are very similar to each other. So 1 photon excited resonance Raman spectrum and 2 photon excited hyper-Raman spectrum are similar to each other. This kind of similarity is also seen about the tyrosine. Tyrosine also has an absorption band here. So when we choose 266 nanometer, by 1 photon, or 532 by 2 photon, we can expect a similar resonance effect. So there was no literature because of the fluorescence. They say 266 nanometer excited resonance Raman of tyrosine was not measured. So let's compare with 240 nanometer excitation. Then large peak appears here. Large peak appears here. And two peaks appears here, 9A and 12. So two peaks correspond to the two peaks. And there are doublet peak. Okay. We cannot directly compare this result and this result because the excitation wavelength is different. But anyway, we get similar information between two photon excited hyper-Raman and Raman spectrum. So here is a list of the 19 amino acids at pH 7.4. So I skip the details, but in summary, in the most cases, the frequency that of the hyper-Raman peak is mostly identical with the frequency that appears in the Raman spectrum. Intensity is different between them. So, at least we should conclude that hyper-Raman band will give unique information if it is large. If the peak is small in the Raman spectrum and large in hyper-Raman spectrum, then it's worth analyzing. But you know, hyper-Raman signal is very weak, and we need to spend long time to get a good result. So one spectrum is measured in 2 hours or 3 hours for each result. If the signal, so if we discuss only the peak position, we do not need to pay much effort to record hyper-Raman. Only Raman is enough. Only when the hyper-Raman intensity is large enough, then it's available to detect some hidden band in Raman spectrum. So in that sense, it's worth trying. That is a tentative idea that I have about amino acid studies. Also, I introduced a few days ago. This is another example of the non-resonance hyper-Raman spectroscopy. So, hyper-Raman signal spectral pattern intensity pattern is different. Peak position is comparable, but intensity pattern is different. So, amide 3 band is large in hyper-Raman, and this is small or not the largest in IR and Raman. So from this result, we see the amide 3 band is strong in hyper-Raman. So possibly suitable for the main chain structure analysis. Amide 3 is related to main chain structure analysis. So if the intensity is large, and it is very small in IR or Raman, then let's try to analyze this weak signal. until now, I'm still pessimistic. In other words, hyper-Raman spectroscopy is thought to be new, but as far as watching the results of the non-resonance hyper-Raman spectrum, it looks like IR and Raman, so not very different. Then how about globular protein? So we finished the preliminary study of the amino acid. So let's try the globular protein. So in this experiment, I picked up bovine serum albumin, BSA. The molecular weight of this protein is 65,000, nearly 65,000. And it contains 27 phenylalanine, 2 tryptophan, 20 tyrosine. But they are only 4.6% of all the number of residues. 2 residues of tyrosine occupies 0.3% of the total length. 20 residue is 3.4%. But they are the aromatic side chains, and actually we have measured resonance Raman spectrum and obtained the information about tryptophan, tyrosine, and phenylalanine. So here is the absorption spectrum, and absorption is at 266. So resonance Raman effect is expected for these aromatic residues. Again, this is resonance Raman. And 266, there is a real state. So this is resonance Raman. Intensity is strong. What happens when we apply hyper-Raman spectroscopy? In this case, we need to consider if there is around 266 nanometer, if there is a 2-photon allowed transition. If we excite the molecule with 1 photon in the normal resonance Raman spectroscopy, we should care if there is a 1 photon allowed transition. In the case of hyper-Raman, we should care if there is a 2-photon allowed transition. And there is a literature, and they say the 2-photon absorption cross-section is considerably large about tryptophan, tyrosine, phenylalanine. So we can expect there is a 2-photon allowed transition near 266 nanometer. So again, we can expect by measuring this protein containing F and W and Y, we could measure resonance hyper-Raman spectrum. Based on this expectation, I measure the hyper-Raman spectrum, 532 nanometer excited hyper-Raman, 532 nanometer excited hyper-Raman spectrum of bovine serum albumin. Result is black line. First, I asked the student, please measure this. Then he showed me this curve, and there is apparently no Raman band, hyper-Raman band. What is this? This is reproducible result, and I don't know what is this. Okay, then let's check what is the appearance of the tryptophan signal, tyrosine signal, phenylalanine signal. Actually, tryptophan, tyrosine, phenylalanine, when we excite with 532 nanometer, large background appears. They give large background. And they are two photon fluorescence. Then by adding these three components, we get a green spectrum as a sum of the two photon fluorescence. Then the fluorescence component almost explains the curved baseline successfully. I should explain. Blue is a tryptophan spectrum. After baseline correction, I got a hyper-Raman spectrum of tryptophan, as shown here. This is after baseline correction. Honestly speaking, much larger two-photon baseline is simultaneously detected in this measurement. But anyway, tryptophan signal is here, and tryptophan signal appears with the two-photon fluorescence like this, and by adding these three curves, we can roughly explain this curve in terms of the two-photon fluorescence of the aromatic side chains. So we can safely subtract this curve as a base, so because we already know the origin. So let's fit by baseline fitting. Then, after subtracting the baseline, we get blue and black spectrum of BSA. So, vertical axis intensity, horizontal axis wave number, and there are some peaks. Okay? Now we can discuss where is the peak by subtracting the large background. And as I explained, BSA, bovine serum albumin, contains tryptophan and tyrosine. So we expect the resonance hyper-Raman signal of tryptophan and tyrosine should be included in this result. So I put two results of tryptophan and tyrosine hyper-Raman signals. Let's compare. The peak marked with blue tick can be attributed to tryptophan. Okay. The peaks with red tick is attributed to tyrosine. Difficult to show. In other words, and some ticks, some black ticks appears only in the BSA spectrum. And they are assigned to the amide 1 or CH bending or something. So, hyper-Raman signal of the bovine serum albumin was explained by only considering the contributions from tryptophan and tyrosine. Okay. Hyper-Raman of BSA, hyper-Raman of tryptophan, hyper-Raman of tyrosine, plus small number of main chain. We found other residue contribution from the other residues such as alanine, isoleucine, leucine, valine, or histidine or some several amino acids does not appear here. Only this result contains the resonance hyper-Raman signal. Here is one explanation. I explained the resonance. I introduced the Raman tensor formulation. And formulation of the Raman tensor contains the terms of the transition dipole moment between ground and excited and ground. Also, it contains the overlap integral of the vibrational wave function. In the case of resonance hyper-Raman, this is a 3-photon process, so 3 terms appear, 3 transition dipole moments, and 3 overlap integrals of vibration wave function appears. In the case of BSA, we should understand like this. In the hyper-Raman process, we are sure there is a 2-photon allowed electronic states. So the 2-photon resonance hyper-Raman signal occurred. Also, as usual, there is a 1-photon allowed electronic level here. So 1-photon excitation and resonance Raman appear. In the case of hyper-Raman process, as shown here, there is no contribution from, or only negligible contribution from the first intermediate state. The resonance effect through upper state, so 2-photon allowed state dominates the signal. In such a case, the consideration of the vibration wave function of the virtual intermediate state can be omitted. So in this case, vibration levels R are summed up, but R vibration wave function R of the virtual intermediate state has no contribution. So we can round up this term to be the unity. Then the overlap integral of the resonance hyper-Raman process becomes this term. And we see it is exactly the same with the resonance Raman term. So in summary, we can say in this way, when the identical and single intermediate state takes part in the resonance, then in such a case, resonance hyper-Raman spectrum is exactly the same with the resonance Raman spectrum. Okay, so this is not good news for us. First, we expect hyper-Raman spectroscopy must be new, and I try to find some new information. But in the case of non-resonance hyper-Raman spectrum, there is not very marked novelty with the result of amino acid. About globular protein, it is shown that resonance hyper-Raman and resonance Raman is exactly the same, theoretically exactly the same. That is, there is no reason we should try resonance hyper-Raman spectroscopy because it's much easier. Okay, still I'm pessimistic. Okay, I add this page. Please refer to this page later. So it recalls your memory about Raman tensor and hyper-Raman tensor. Okay, but we need to continue the studies of biomolecules. So next target is the nucleic acid. Nucleic acid is a building block of the DNA and RNA, and 4 kinds of the building block A, T, G, C are known about the DNA. And they equally have absorption at 266. So again, we expect resonance hyper-Raman. Okay, with 532 excitation. 2-photon process of 532 nanometer. So it resonates to 266 nanometer absorption. Okay. In the previous case, resonance hyper-Raman spectrum was exactly the same with the resonance Raman spectrum. So already I'm pessimistic. And about in the case of DT, DG, DC, so the model compound related to thymine, guanine, cytosine, 532 hyper-Raman spectrum upper was almost exactly the same with the 266 nanometer resonance Raman as shown here, DG, DT, DC. Okay, so briefly speaking, resonance hyper-Raman spectrum is again the same, identical to resonance Raman spectrum, not new. So from this result, we can say this is the 2-photon resonance hyper-Raman spectrum with 1 electronic state. So 2-photon process and 2-photon resonance Raman and 1-photon resonance Raman is resonated to same electronic level. Then it becomes identical. So at least we can conclude resonance condition is satisfied by the same electronic state. Okay, that is some conclusion with 1 electronic state. Identical electronic state is in charge of the resonance effect in the 2 different processes. But again, not different from resonance Raman spectrum. Okay? Please be patient. I have been patient for a few years. So continuing amino acid and protein and nucleic acid. Okay, no difference, but let's continue. And in the case of adenine, DA, eventually we found some difference between 2 spectra, 532 hyper-Raman and 266 resonance Raman. Okay, some peaks selectively appears in hyper-Raman. And this peak also. And this peak also. So in such a case, new information is obtained only from hyper-Raman spectrum. What is the difference between this case and previous three cases? In the case of DA, it is known that there are two electronic states, so-called LA and LB, near 266 nanometer. And the nature of these electronic states have been studied in detail with the computational method. And this difference is explained by taking into account the difference in the character of the two electronic states. One should be one photon allowed. The other should be two photons allowed. Exactly saying, the symmetry of this molecule is not so high, only there is a symmetry plane. And therefore, one photon allowed state and two photon allowed state are not exclusive to each other. So this is large absorption with one photon and small cross-section with two photons. One state should have such a property. The other state, large two photon absorption cross-section and small one photon absorption probability. Then, using one photon excitation, this process should resonate largely to one photon allowed state and weakly resonate to the small one photon state. On the contrary, two photon process should resonate to large two photon allowed state and minorly resonate to the small two photon state. So briefly speaking, the significance of the two states is different in the resonance Raman and hyper-Raman process. Then the resonance effect due to LA and effect due to LB becomes different. So in the case of a hyper-Raman and resonance Raman, different feature appeared. Okay, please recall, let's get back to globular protein. I explained when identical and single intermediate state is in charge of the resonance, then they are identical. If there is only one electronic state and one photon allowed, two photon allowed, the same and only one intermediate state is present, then they become identical. But we found that if there are two electronic states, then the intensity pattern becomes different. So from by analyzing the intensity difference or some peak position and intensity of the new peaks, we could be able to study the contributions of the two intermediate states. In this regard, this hyper-Raman spectroscopy is a new approach to study the resonance enhancement process. Okay. First, I found some new information using hyper-Raman spectroscopy. Okay. So this kind of analysis is now going on. By the way, I compared the hyper-Raman spectrum of DNA polymer. This is the result of DNA by using salmon DNA. Then compared with the hyper-Raman spectrum of the mixture of A and T or G and C. Then I saw the complete corresponding relationship of this result and linear combination of these two results. That is, hyper-Raman spectrum of DNA is explained by considering the spectra of ATGC. Okay, so I wanted to show when we apply the hyper-Raman to DNA, some new information should appear. So new peak appears that is not explained by the linear combination of these four, but in vain. It was impossible. All the peaks were explained by the linear combination of these four. So in this meaning, it's the same with using resonance Raman spectroscopy. Okay. Then anyway, let's try biomolecule, sugar. And this is surprisingly new result, honestly saying. Okay, so biomolecules. So sugar is very important. Our cell is contains a nucleus or several organella, and it is covered by lipid bilayer. And lipid bilayer, it is known there is a lipid bilayer, so this is lipid. Then what is the surface? Actually, there is many sugars saccharides that covers the cell surface. So it's very important to study sugars and using vibrational spectroscopy. But organic synthesis of sugar is still not easy because this is monosaccharide. If there is another sugar comes, this OH, this OH, this OH, this OH, all reactive. So there are too many combinations of the binding, even if we consider disaccharide. So because of this variety, still the organic synthesis of sugar is not very easy. And that is one of the reasons the study of the sugars using vibrational spectroscopy has not been done, in particular about the long chain. But anyway, so for that reason, I tried to analyze the sugar by using hyper-Raman spectroscopy. I wanted to use oligosaccharide, but first let's start from monosaccharide, glucose. This is the simplest sugar. okay, building block of the polysaccharide. So I dissolve glucose into the phosphate buffer serine at pH 7.4. Okay, let's measure hyper-Raman spectrum. Then we observe the blue spectrum. At a glance, it's very strange. Why? Peak appears above 1500 wave number. This molecule consists of C and H and O. So if you have experience to analyze the vibration spectrum, you may know there is not so many residues functional, not so many functional groups give the peak above 1500. Some functional groups are known: nitro group NO2, NH3 plus, and CO double bond, CC double bond. But you know, there is no CC double bond, there is no nitro group, there is no NH3. There is no reason peak appears above 1500. First, I asked the student, did you do wave number calibration properly? And then he replied, yes, I did, as you taught. Okay. Then I checked, and it was reproducible. So we have to consider what is the origin of this molecule. These peaks. And green is the Raman spectrum excited with 532 nanometer or 266 nanometer. Peak position is identical between these two results. So that is, they are the non-resonance Raman spectrum. So this molecule does not have the chromophore at 266 nanometer. So even we use UV excitation, it is non-resonance Raman. And again, as expected, all the peaks appear below 1500, as expected. We know CH bending band appears near 1400 or somewhere. CC stretching appears around here. CO stretching appears here, or CC appears. Or until now, all the peaks are already assigned to some molecular vibration. So these two are acceptable result as a Raman spectrum of glucose. IR spectrum yesterday, 2 days ago, we measured, and it gives the peak. And it is again already assigned peaks. So IR and Raman spectrum are measured properly. We have a good skill already. But hyper-Raman signal is quite different. What is this? And more strange, in the phosphate buffer serine we detect signals, but when we remove phosphate buffer and NaCl, that is, when we dissolve the glucose in H2O, then the signal disappears. It's quite new, surprisingly new, honestly speaking. That is, peak position in the hyper-Raman spectrum is not identical to those in the IR and Raman. Intensity pattern is also different from IR and Raman. So this is a unique, very unique information using hyper-Raman spectroscopy. Then finally, I was glad. I can say hyper-Raman spectroscopy gives a unique information. But we don't know what is the origin of this peak. So we started to study the origin of this hyper-Raman signal. So this is the result of glucose, and we chose this molecule as a fundamental sugar molecule, smallest sugar molecule. In the sense of the biology, it is smallest sugar. But from the viewpoint of chemistry, it is still large enough. Okay, many 6 carbons, 6 oxygens, 12 hydrogens. It looks too large. So let's pick up the model compound. So we chose trifluoroethanol as a model compound because it carries CH2OH group, and this is also seen in the sugar. So let's see what happens on this part. Then we measure the 532 nanometer hyper-Raman spectrum of trifluoroethanol. And again, we found the peak above 1500. Okay, this is a good signal. So it's worth discussing this molecule as a model of the glucose. But in this case, the signal appears at extremely basic pH. At neutral pH, signal does not appear, and only at the basic pH, similar signal appears. In PBS, the same signal appears, but to simplify the solution, we chose the basic condition to study this signal more. Again, the hyper-Raman spectrum pattern is completely different from the Raman signal. And Raman signal are measured at pH 7.1, 7.0 and 30.2, and spectral pattern is similar. So even at a highly basic condition, this molecule is not decomposed because the Raman signal is fine. So this is not a decomposed compound. So first information was that this signal was observed at basic condition. So instead of considering this structure, let's consider trifluoroethanol anion. Next, we check the laser power dependence. As we know, hyper-Raman spectrum intensity is in proportion to the quadratic power of the incident power. So we expect I square dependence. Actually, when we analyze the intensity of the hyper-Raman signal of H2O, it exhibits quadratic dependence. So laser power is changed from 141 to 212 milliwatt. Then the intensity gradually increases. Okay. And now we are focusing on the signal of TFE, and you see in the beginning there is no signal, but it appears and suddenly grows up. That is, power dependence is different from typical hyper-Raman signal. That is shown here. Expected power dependence is slope is something like this. And the hyper-Raman signal of TFE has sharper dependence. That is I to the 8.8 plus minus 0.6 power. From this fact, we concluded this is not hyper-Raman signal of the stable species. If this is originates from the stable species, power dependence should be the quadratic, but it is quite different. So this must be the result of the photoreaction. Then I looked for the literature, and one possible explanation was the 3-photon excited radical formation of H2O. So they say when we irradiate 172 nanometer to H2O, it formed radical, OH radical and hydrogen radical. So we adopted this idea. At least there must be a photoreaction, and possibly it is the radical formation reaction due to the 3-photon process. Anyway, we need to explain such a large number dependence. So 3-photon dependence is explained here. Okay. So let's update the picture. A plausible model should be not only the anion, but also radical anion of trifluoroethanol. And next experiment is the concentration dependence of TFE. Then signal intensity changes as the concentration changes from higher to lower. And there was a clear difference of the spectral pattern depending on the concentration. At the highest concentration, intensity pattern is something like this. Right-hand side is stronger, nearly twice or 3 times stronger. But in this case, intensity is comparable. Also, as the center position is here, indicated by red line, but as concentration increases, the peak position shifts to lower. So we found the intensity pattern was concentration dependent. We have to conclude there are multiple species. So by analyzing the data set using some mathematical way, we found this result contained 2 independent components. And it was shown to be these 2 components having black spectrum and black concentration dependence, the component having blue spectrum and blue spectral components, concentration dependence. There are 2 components. And by referring to some literature, we concluded this curved species should be monomer radical anion. Linearly increasing component is dimer species. So we tried to, we used a trifluoroethanol, but there was a dimer formation. So updating the picture, monomer radical anion and dimer radical anion should give these signals. Then we employed some computational calculation. Okay, so by using a DFT calculation, we got some information about electronic states and vibration frequency. We tried examine the 5 species: neutral TFE, anionic TFE, TFE radical anion, TFE dimer anion, TFE dimer radical anion. As expected, if the TFE exists in the neutral form, then the absorption band or electronic state is all below 200 nanometers. As you may know, TFE is a transparent liquid. There is no visible absorption. It agrees with this calculation. Okay? And by forming the anionic form, distribution of the electronic states becomes broader. Radical anion has broader distribution. It's also the case for the dimer anion and dimer radical anion. Radical anion has a broader distribution. And here, let's see the vibration frequency. First, I felt this result was curious because the peak appeared above 1500 wave number, where only the limited number of the functional groups give the peak, NO2, NH3 plus, or C double bond, CO double bond, and so on. It was impossible for glucose to give the peak in this region. And it is also impossible for trifluoroethanol neutral form. But you see, when we consider the radical anion, then molecular vibration, these two structures has a molecular vibration in the range above 1500. So radical anion is CF3 CH. This is the neutral form, and radical anion is formed when H is removed and it becomes anion. And H is removed by the radical reaction. So radical anion of trifluoroethanol have CHO group. And the CO stretching of this group gives a peak above 1500. Okay, so this consideration allows us to assign an unidentified peak above 1500 to be part of the radical anionic form of the CHO group, trifluoroethanol. But do you remember? This form was not detected in IR spectrum or Raman spectrum. If this species exists, why it is not detected in Raman spectroscopy, IR spectroscopy? That is a question. To explain that point, let's focus on resonance condition. When we check the electron distribution of the electronic states of these four species, anion, radical anion, dimer radical anion, all have the electronic state near 266. So they have a chance to give the resonance hyper-Raman signal. Okay? So their signal should be enhanced more. But it is the same with resonance Raman. One more thing we should notice. Here is the electronic state near 532. Okay, hyper-Raman process is a two-photon process. Just a moment. Two-photon process and the electronic state near 266 nanometer allows resonance hyper-Raman effect 532 by 2 photon. Okay, and only in the case of radical anion, there is real state near 532. That is, these molecules undergoes 1 photon resonance and 2 photon resonance, double resonance. If we consider in this way, it's plausible that only these species give the extremely strong signal, hyper-Raman signal, because they undergo the double resonance, 1 photon resonance, 2 photon resonance. So if we expect 10 to 6 times enhancement with a resonance enhancement, in this case 10 to 6 by 10 to 6, 10 to the 12th enhancement. By considering in this way, we can explain why this species only appears in the hyper-Raman spectrum. In the IR and Raman spectrum, their signal is too small to detect. Other species, neutral or anionic species, gives larger signal, so their signal is hidden or undetectable. But thanks to the double resonance enhancement, their signal is extremely largely enhanced, and then it appears in the hyper-Raman signal selectively. As a consequence, their signal is much larger than the other species. So the diagram is here. TFE radical anions monomer and dimer undergoes 1 photon resonance because there is a real state, and 2 photon excitation and there is a real state. So their signal is doubly enhanced by the resonance effect. This effect is not available in the IR spectroscopy. It absorbs IR light and 1 photon excitation occurs. Or in the Raman spectrum, resonance Raman spectroscopy, 1 photon excitation and 1 photon emission. In the case of resonance Raman, only 1 resonance effect is available. In the case of hyper-Raman, twice double resonance is available. So it's a quite unique feature of the hyper-Raman spectroscopy, and I believe hyper-Raman spectroscopy can be a method to detect a very small number of molecules by utilizing double resonance effect. Okay, so so far the discussion is about the trifluorescent or TFE radical anion. But we started the discussion from the glucose. Okay, so it's time to return to the glucose signal. Okay, again, it is about 1500, and probably it is not a vibration of the glucose itself. We can expect it should be the radical anion of glucose. In support of this idea, we have observation in PBS signal appears in H2O no signal. That is, phosphate buffer sharing should have some role. And I found literature. Phosphate ion captures radical and it becomes phosphate radical and OH minus. This experiment is done at pH 7.5, 7.4. So it's neutral. But once OH radical is formed, then it turns to phosphate radical and OH minus. And OH minus lowers pH at least very locally. And radical reaction is enabled by this guy. So even at a neutral pH, following the radical formation, some radical anion formation of the glucose should take place. Phosphate radical and OH minus generate the glucose radical anion. This is a possible story. And again, glucose radical anion is selectively detected. Other peaks of glucose are not seen. In particular, in this region there is no signal, even though Raman and IR give the many peaks. That is, this species is selectively detected in hyper-Raman spectrum. It's because double resonance effect enabled the extreme enhancement of the hyper-Raman signal. So the intensity of this species became much larger than the remaining species. That is the explanation at present. And this is a kind of recent progress. Okay, so we started a study of glucose and obtained this result I introduced in the previous page. And already we moved to other many sugars, and irrespective of the sugars, they give almost the same intensity pattern. So probably origin should be common among them. Okay, so now I'm thinking about trying to elucidate what is the common structure. It may be possibly the radical anion, and I want to know which sugar is most susceptible to the radical attack. That is related to the damage of the sugars in our body. Okay, in our body, radical is formed, and it is eliminated by some proteins or vitamin C or some compound. But by chance, it will damage sugar, and it makes radical anion sugar and changes the structure. Then I suppose that kind of radical reaction should be related to the dysfunction of the human body. So for that direction, I want to study sugar dependence and check the susceptibility quantitative analysis. This is now underway in the laboratory. Okay, so this is the last page, summary, hyper-Raman spectroscopy. What is new in biomolecular studies? Okay, in the beginning it was pessimistic. In the case of non-resonance hyper-Raman, intensity pattern is different, but peak position is the same. So if large peak is very important, then it's worth trying to measure hyper-Raman. Otherwise, information should be similar with the Raman spectroscopy. When we measure the resonance Raman spectrum, if the intermediate state that is related to resonance condition is identical between resonance Raman and hyper-Raman, then it is theoretically shown that the spectral pattern is exactly the same. So this is not new. Okay? If there is a multiple intermediate state, intensity pattern would be different depending on the contributions of these two states in each process. So this is okay, worth analyzing. And in the last example, if some species is allowed to resonate to two intermediate states, then the double resonance hyper-Raman is possible. And in this case, very unique information will be obtained from the hyper-Raman spectroscopy. Okay, this is a summary of the novelty of the hyper-Raman spectroscopy. So depending on the condition, information may be new or not new. Sorry, I prepared some. Okay, if the intensity is different, new marker band may be found. There is no new point. In this case, resonance enhancement process and information about the intermediate state. And double resonance hyper-Raman allows us to detect the minor species. Okay, so this is the summary of this talk. Okay, so this is all.